Hello everyone, welcome back to Chinmoy's Biology channel to a yet another new episode of Polygenic Inheritance Part 2. So in the previous episode, we have already spoken about the many important things and basics. So if you have not gone through that, I'll be putting the link in the description box. So please do go through it first. Now in today's episode, we will be going to discuss about the main important examples of polygenic inheritance, like the color of the wheat kernel, about the skin color, about the height of human beings. So these are the things we are going to discuss today in details with notes and with diagrams and with obviously the genotypic forms and all. So please do stay tuned throughout the full episode. Don't skip in between, because if you are skipping, you will be missing many important topics of discussion. So please keep staying with us throughout the episode. So let's start with today's episode of Polygenic Inheritance Part 2. So first we are going to discuss about the polygenic inheritance in humans, right? So in humans, there are many traits that show the quantitative inheritance. So we have spoken in the earlier episode that this polygenic inheritance is also called as quantitative inheritance, right? So some of the examples are skin color, eye color, height, and risk of certain diseases, right? So which are genetically inherited from the parents to the offsprings. Now first, when it comes to skin pigmentation in human skin color is a polygenic trait or a character. It depends on the presence of the melanin pigment, and the amount of melanin produced depends on the three pairs of genes A, B and C. So these are the genes which are producing these three pairs of genes are producing the melanin. Okay, so on that melanin production only the skin color of human body depends. So these genes are present on different loci, and each dominant gene is responsible for the synthesis of a fixed amount of melanin. Is it clear to you all? So each dominant gene is responsible for a fixed amount of melanin secretion. So the effect of all the dominant genes is additive. So they all together. So when we add the effects of three genes, additive, the effect of these genes, they will show you the dominant trait. So thus the amount of melanin produced is always proportional to the number of dominant alleles. So all the dominant alleles together, what they are producing melanin is a combined effect. It's an additive effect. Okay, so that is what we have spoken earlier also. The same thing we are insisting again and again. So it should be in your mind and you'll not forget it. Okay, the homozygous dominant allele are responsible for the dark phenotype, and the homozygous recessive allele are responsible for the light phenotype. So when black or extremely dark, that is AABBCC. Okay, so pair of genes, three genes, that is the extremely dark phenotype, and this they are showing the extremely dark phenotype, and white or albino is recessive A, recessive B and recessive C. So people marry the offspring, people marry, and the offspring or the individuals of F1 generation have a mulatto-pigmentation, that is a mixed or a wheatish complexion they'll have, that is a mulatto-pigmentation. Okay, so that in details in the next slide we are going to discuss. When this person is marrying this person, so what is happening in this F1 generation, what is formed? So what is happening if this dominant parent is marrying to this recessive parent having this three genes? This is dark, and this is we can say this is light skin tone, right? So when they are getting married, so when they marry each other, so in F1 generation the intermediates are formed. So they are having a combination of all these two genes. So they are intermediates. So they are neither too dark nor too light. Now what in F1 generation this is happening? Now as in Mendel's cross, we know that selfing is done. Okay, so with this again selfing is done. So what is the result of this selfing? So what we will get, that let us see that in the next slide. So after that selfing, we are getting this. So the gametes, male and females, will be like this. So we are getting the gametes like this. All combination of gametes will be male and female like this, and we will get this Punnett square. So this is a quite difficult thing, but it is actually not at all difficult. You know that when you are forming the gametes, you know you are just multiplying this. So A, B, B, C, C. So when you are multiplying this with this, you will get capital A, small a, capital B, capital B, and capital C, capital C. Okay, so we should just see the combinations and we can find out that whether it is dark or whether it is fairly dark. So if they are having two dominant genes and one combined heterozygous gene, so that will be dark. If they are having two recessive genes, that will be partially fair, fairly dark, and also so many combinations here it's there. So what are all the phenotypes we are getting in this? Actually, this is not at all for your class. You please, for 12th standard, it is very difficult, but for your knowledge in other exams such as NEET or any AIMS exam, so you can get a clear idea about what is happening if you are forming the gametes from the previous F1 selfing. You can get these gametes, and if you are placing this gametes as male and female, you can get this Punnett square. So here when you are completing the Punnett square, you will see that only one is there which is very dark. Okay, then comes 6 which are dark, fairly dark. 15 combinations are there genotypically. Intermediate is 20 and fairly light is 15. So what we are seeing, the phenotypic ratio is 1:2, 6:2, 15:2, 20:15. 15. So this is the phenotypic ratio of this F2 generation. So this is what will be visible among the offsprings. So they will be one very dark, one fairly dark, one intermediate, and one fairly light. So this is the Punnett square. So let us see how many combinations are being formed here. So in the gametes, there are a total of 8 combinations, okay, resulting in 64 genotypes. So 64 genotypes will be there, resulting in 8 combinations, very dark or pure black, that is 1 in 64. 6 in 64 will be dark, what we have told in the phenotype, because 64 is the genotype. 64 type of genotypes are formed. Fairly dark are 15 out of 64, intermediate is 20 out of 64, fairly dark is 15 out of 64, light is 6 out of 64, and very light is 1 out of 64. So very dark is 1, very light is 1. So this is what is the human skin color. So now we will move on to the height. Now, as per the ratio, as per the phenotypic traits we have seen in the color of the human beings, you can see the diagram over here. This is very dark one, and you can see that among this is very light one. So these are other different color, other color combinations of the that experiment which we have seen. So many types of color variations, skin color variations are there, and which is a very good example of polygenic inheritance of the human skin color. Okay, so you can see so many variations are there. One is very dark and one is very light. Other than that, there are a huge lot of variations. Right, now we will move on to height. Now, height in a man is also controlled by 3 pairs of genes located at different gene loci. So in skin color also 3 pair of genes were located in the 3 different gene loci. Okay. So the height develops by the additive effect of number of dominant alleles of these genes. Same, they are having an additive effect of the dominant alleles, and people with all the 6 dominant alleles are the tallest, and with any 3 dominant alleles are average height, and with all recessive alleles are no dominant alleles are shortest. So same as that of the white skin color, dark skin color, and all. With all light skin tone, they will have all recessive, like that. The shortest will have all recessive, the tallest will have the 6 dominant alleles, and who are intermediate will have 3 dominant and will as well as 3 recessive, right? Now, in this diagram, we will discuss what we have spoken earlier, that is the quantitative character of height that in polygenic inheritance. So, all the dominant characters, if they are present, all the dominant genes, it will be the tallest one. All the recessive alleles are present, 6. So, it will be the shortest, and within that, all which are present are the intermediate one, having the medium or the average height. So here you can see so many variations of height are there in humans. So one will be the tallest and obviously one will be the shortest, but there are a wide variety of height in all human beings, right? So let us move on to the next one. So the eye color in humans is another example. So this is a bit difficult part in the next to these two parts. So no need to learn or memorize that. Just for idea of this, I have written this. You just go through it once. Okay, the color of the eye is determined by the polygenes. We all know, like the previous ones. It is identified that a minimum of 9 colors of eye are recognized in humans. So there are two major eye colors genes and 14 more that normally determine the expression of phenotype in human beings. So a different number of alleles contribute to each color. So 2, 3, 4, they are contributing to each color. The eye color is mostly associated with two adjacent genes on chromosome 15. So where they are present on chromosome 15. Two adjacent genes of chromosome 15, they are associated with a human eye color. So the OCA2, that is oculocutaneous albinism type 2, and HERC2 gene are mainly associated with eye color. So oculocutaneous albinism type 2 and HERC2 genes. HERC2 gene is a giant E3 ubiquitin protein. So these all for higher classes it's there. I'm just writing it to make you you can have a clear view of what is happening and just to know the names. So it is a protein ligase which is responsible for the pigmentation or the eye color. Okay, so let us see what are the different types of eye color. So here you can see a wide variety of eye colors from blue to brown to wheatish. So many types of blue shades are only there. They are dependent on polygenes. We have spoken about 2-3 genes there. So many other genes are there. Mainly the two genes present on chromosome 15 are responsible for the variety of eye colors. So variety of colors are there in this slide you can see. So next we will move on to the plant polygenic inheritance. So polygenic inheritance is also seen in plants. Some examples of polygenic inheritance are listed below. What are that? Color of the stem, shape of the stem, flowers, pollen grains, oil content and yield. These are all the, we can say they are all dependent on the polygenic inheritance. So many genes are showing these phenotypic characters or traits. Okay. So the kernel color of wheat was the first experiment that was done to find the evidence on this polygenic inheritance. Okay, so what the color of the wheat is controlled by two pairs of alleles. They show independent assortment, right? The dominant genes are R1 and R2 and the recessive genes are small r1 and R2. The homozygous dominant allele, these both are cumulatively effect is they are responsible for the dark phenotype or dark red color of the wheat kernel, and the homozygous recessive are responsible for the light phenotype or the white color of the wheat kernel. So for example, the dark wheat will be R1 R1, R2 R2 is crossed with the white wheat kernel, that is R1 R1, R2 R2. So in F1 generation it is obtained an intermediate red color, that is capital R1 small r1, capital R2 small r2. Okay, so that is an intermediate red color, neither white nor dark red color. So the F1 generation is again self crossed as in Mendel to obtain the F2 generation, and the F2 generation produced 63 red kernels and with different red shades and one white kernel. So this 63 red kernels had different variations. So they were different. So different variations in red had happened. So that showed that there is a polygenic or a cumulative effect of these genes on this kernel color of wheat, and only one white kernel was there. Is it clear? So let us see the diagram of the wheat kernel. Now let us see, this is the parent kernel, this is capital A, capital B, capital C, small a, small b, small c. So there is a dark red kernel, wheat kernel, and the white kernel, which gives after crossing, which gives to the intermediate red, or capital A, small a, capital B, small b, and capital C, small c. Now in the F2 generation, after selfing, what we are getting, same, we will get 1 dark red, 6 moderate red, 15 red, 20 intermediate red, 15 light red, 6 very light red, and 1 white. So same as that of human skin color and all. Okay, so 63 red shades are there and only 1 white shade is there. So there is a 63:1 ratio, okay, phenotypic ratio, where 63 are varieties of red shades and only 1 is white, and this is the polygenic inheritance of wheat kernel diagram. So this is all in polygenic inheritance, what you are having in your 12th curriculum CBSE. So it's all covered with most of the important parts which will be there in your NEET as well as in other competitive exams which you will be sitting. So go through it in details. I think if you are having any doubt, you can ask me in the comment box, and if you are not getting in once, you can repeat and rewind the videos twice or thrice, and please try to understand the concepts with the notes and with the diagrams which I have shown you. Still then, if you are having any doubt, please do write in my comment box, and please stay tuned, subscribe to my channel, keep watching, because the next video will be on pleiotropy, which is another important topic, and stay tuned for many more episodes and interesting topics as per your curriculum. Thank you. Thanks a lot for watching.